Okay, so then part of the special senses is also the sense of hearing, which is conveyed by the ear. The ear is subdivided into three segments. The segment that is external to the auditory tube, to the tympanic membrane, which is called external auditory canal. And the function of this, in general, is to collect the sound waves that we have in the environment and to bring it into the tympanic membrane, which is a very thin membrane that vibrates and that is connected to the auditory ossicles in the middle ear. So the middle ear is located between the tympanic membrane and the round window and the oval window. And that's where we have from lateral to medial, MIS, malleus, incus, and stapes. These tiny ossicles, they amplify the vibrations received by the tympanic membrane, and then they transmit this into the couple of membranes that we have, into the basilar membrane that we have inside the inner ear. And then in the inner ear, which is just beyond this oval window and the round window, it has a structure that is called the vestibulocochlear apparatus. In the vestibulocochlear apparatus, you have the vestibule, which is where you have the cells that sense the movement of your body, and then it sends signals, actual potentials, into different centers in our brain, and then those centers in the brain, they send efferent signals into the muscles of our body so that we can maintain equilibrium, we don't fall. Also, these action potentials and this transfer of information given by these action potentials tells us the characteristics of the velocity as we are moving, like how fast you are moving, as well it tells us the direction of the movement. Okay, so we have several information coming from the vestibule, which is part of the vestibulocochlear apparatus, and then we have a part that is called the cochlea. The cochlea, which is connected to the cochlear nerve, it helps us to hear. So then we have this apparatus that it is in charge of hearing and balance. Let me change it here. So here is the location of the three parts of the ear. Anything external to this membrane, which is the tympanic membrane, will be external ear. Anything here in this tiny region, it is the middle ear, and anything here will be the inner ear. So, the external ear is made by the auricle or pinna, the lobule, and then this canal, which is partly made by bone, the temporal bone, and partly made by cartilage. And then here, at the entrance of this, we have ceruminous glands that produces a waxy material that helps to prevent the entry of debris into our ear and also insects. As well, it has acids that helps to prevent the growth of microorganisms. And then here we have in the middle ear the malleus, which is connected to the tympanic membrane and also connected to the incus. And then the incus is connected to this stapes that has a plate that covers the oval window. So as the vibrations are happening, this stapes and its plate goes out and in, out and in, out and in, closing and opening this oval window, allowing the transfer of vibrations into this vestibulocochlear apparatus, which is part of the inner ear. This one is completely embedded within the petrous part of the temporal bone, and this apparatus has this part that is called the vestibule, where you have the oval window, the round window. And this round window, what it helps to do is actually, as the vibrations and the pressure is transferred from the middle ear into the inner ear, it will allow part of this pressure to go out so that you don't have as much pressure accumulating in this area. And then we have these semicircular canals. We have one that is anterior, another one that is lateral, and another one that is posterior. That are the ones that help us to sense the movement of our body, especially the head. So if your head looks up, if you're just facing anteriorly, or if it looks down, and also if you move to the sides, if you tilt your head. So all of this is given by this. And also it tells you about angular acceleration. And then this part, the cochlea, is the one that helps us to hear. So we will see internally what the cochlea has. And of course, connected to this vestibule is the vestibular nerve, and connected to the cochlea is the cochlear nerve, that both of them together they form cranial nerve number 8, vestibulocochlear. This is part of the special senses nerves. So remember, we have 3 cranial nerves that are part of the special senses. Olfactory for smelling, 2 for for seeing, and 8 for hearing and maintaining balance. And then, so we have air here in the outer ear, we have fluid in the middle ear, and then we have fluid in the inner ear. And now we have a connection between the middle ear and the pharynx, which is called pharyngotympanic tube, or also auditory tube, or eustachian tube, that helps to basically equilibrate the external pressure with the internal pressure, so that whenever we are changing altitude, then that pressure, that as we go into higher altitude, doesn't burst our ear. So this one closes as you move up. It's made out of muscle, this pharyngotympanic tube, and then if you chew gum or if you start yawning, you will be able to open this pharyngotympanic tube, and then you will be able to hear better. Okay, so that's what happens. So then how hearing is processed? So first, you will have sound waves in the air that travels as a sound wave, and then it hits your tympanic membrane. So these are collected by auricles. And then the tympanic membrane vibrates. That makes the ossicles to vibrate. And then the oval window opens and closes as the stapes moves in and out of the oval window. And then that transfers these vibrations into the basilar membrane of the cochlea. And then in the cochlea, we have the organ of Corti, or spiral organ, that basically is made out of hair cells. And as these hair cells vibrate, they touch like a tent-like membrane that's called the tentorial membrane. And that is going to lead to the polarization of these cells, and then transfer of action potential through the cochlear nerve into the auditory cortex, which is located in the temporal lobe for perception of hearing. So then, this is what happens with hearing. So we have sound waves that are represented by these lines. This will be the external acoustic meatus, or auditory canal, that hits the tympanic membrane. Auditory ossicles vibrate. That creates pressure waves that goes here into this membranous portion of this vestibulocochlear apparatus. So this is looking internally to here. So we have then this bony portion of the apparatus. This is like if you extend it all the way through. Of course, it is all folded in like if it was a snail. So this part, it is called the helicotrema, the tip. So this is the helicotrema extended and also the foldings. So then when this oval window is open and closed by the stapes, you create pressure waves. And these pressure waves migrate through some lymph that is called perilymph in this upper part of this membrane. So this is the basilar membrane. And then here, this middle region, it is called the cochlear duct or scala media. This will be the scala tympani above the scala media and the scala tympani below. So the scala that it is connected to the oval window will be the vestibuli, and the one that is connected to the round window will be the tympani. So then as these waves of pressure migrate, you will have also movement of this part. So if you just take one part out, you will see in the basilar membrane that here in this region, in the proximal end, you have this portion that is narrow and is stiff. So it can help us to hear a unit of hearing of waves, which is called the hertz. So over here, this can receive basically a number of vibrations or waves that are 20,000 hertz. And then here, next to this part, to the distal end, here is very wide and flexible. So this one, it can have sound waves of 20 hertz. So if you have a lot of vibrations that are close by, like in this case 20,000 hertz, this is called high frequency sound waves, and then in here will be low frequency waves in this region.
So is that like the pitch of sound?
Mm-hmm.
Low pitch and low pitch?
Yes, so it's related to that, the low pitch and the high pitch. And then these waves will be transferred. And then as they transfer, you will have embedded within this area the organ of Corti with the hair cells that will depolarize. And then that will send the action potentials into the cochlear nerve. And then whatever pressure it is in excess, it will exit through this round window. So then everything is just transferring pressure from middle ear into the inner ear. High frequency sound waves will be detected in here. Low frequency waves will be close to this helicotrema. So high frequency wave, low frequency waves there. So now if you magnify one of the regions within this cochlea, so you will be able to see the different parts. So this is the scala media, above the scala vestibuli, and below the scala tympani. These two has a fluid that is called perilymph. This one has a fluid that is called endolymph. They have different contents of ions, and then endolymph is more like our plasma, the content of ions. And then for this basilar membrane, which is found in this scala media, we have here some hair cells, which are mechanoreceptors that will detect the change in pressure, and then they will start moving like this. And then on the tip of these hair cells, you will have this cilia that will touch this membrane that is called the tectorial membrane. As this touch this tectorial membrane, you will have depolarization of these cells, and then these cells will generate an action potential that is going to be transmitted through these axons that are going to reach the spiral ganglion. Then you will have a synapse with some cells here, and then these will extend the axons to form the cochlear nerve. So this is the magnification of this area. So this is the organ of Corti, or spiral organ. So if you magnify these cells, so here are the hair cells, and these cilia will be some short and then one that is long. So they progressively increase in length, these cilia, on the top of these inner hair cells. So here's the nucleus, and then these hair cells have synaptic vesicles that are going to be released, and then you will have activation of this cochlear nerve axon. Now, this stereocilia is linked with these fibers. So then, when you have sound waves, so this is the basilar membrane, this. So when you have sound waves, then you have bending of these stereocilia. So here you can see these micrographs, and then here are the links of these stereocilia. So these ones will vibrate, and then they will bend, and then when they bend towards the tallest stereocilia, the longest, you will have opening of sodium channels. So there is like a physical connection of this with the sodium channels. So then the sodium channels open. That will help for depolarization of these hair cells, and then you release these neurotransmitters that are going to lead to the depolarization of this neuron, of the cochlear nerve. So then these action potentials from the cochlear nerve will go and then send these action potentials into this nucleus, the superior olivary nucleus and the cochlear nucleus, via a synapse or directly. And these will be located in this area just above this cochlear nuclei, and then these cochlear nuclei will extend axons to synapse with the inferior colliculus of the corpora quadrigemina. So then these inferior colliculus receive auditory stimuli, and then the superior colliculus, if you remember, they receive visual stimuli. Now, from this part, then this inferior colliculi is going to extend some neurons that are going to connect into the medial geniculate nucleus in the thalamus, and then from there you will have axons that extends to the temporal lobe in the auditory cortex, in the superior portion of the temporal lobe, which is this. And this is where we have the primary auditory cortex. That's where we interpret the sound. So as you can see, there are several neurons. So we have one here between the cochlea and the cochlear nuclei, which is located in the pons. And then we have the second neuron between the cochlear nuclei and the superior olivary nucleus. And then we have a third axon, or neuron between the superior olivary nucleus and the inferior colliculus. And then, so we have 1, 2, 3, and then 4 one between the inferior colliculus and the medial geniculate nucleus, and then 5, one between this and the primary auditory cortex. So then with this sense of hearing, you have greater chances of basically having something going wrong because you have 5 different type of neurons. But that's the neural pathway. Okay, so now for how the vestibular system maintains equilibrium. So it's similar to hearing in a way that we are using mechanoreceptors. So we have an area that is called macula, just like as the macula lutea that we have in our eyes, but in this case, this macula will detect a static equilibrium. So basically, will be the head position relative to gravity. When you are static. Now we have within the semicircular canals these hair cells that detects motion, head rotation specifically. So you can tell whether your head is rotating to the sides, or if you are just looking straight ahead, or if you just tilt your head. So that is detected by some cells that are called the crista ampullaris, where you have these hair cells. And then these mechanoreceptors act by having a gel-like portion on top that it is filled with calcium salts. And then when this this movement happens, then these stereocilia, or these cilia in the hair cells, can move this gel, and then you can detect what type of movement you have, and then you will adjust your body to maintain equilibrium so that you don't fall. And then same as for hearing, the cranial nerve number 8 is going to receive this information through this vestibular nerve, and then you connect to the brainstem and the cerebellum to maintain balance and coordination. So here we have some examples. So this gel-like substance is called the autolytic membrane. And it is not shown in here, but we have a lot of calcium salts. So here are the hair cells, and then we have these cilia that are called kinocilium, that it's embedded specifically within this autolytic membrane. So then if someone is not moving their head, is just completely looking directly, you will have these hair cells constantly releasing a particular amount of neurotransmitters into this vestibular nerve, so that you have a particular intervals of time that happens between these action potentials. So these action potentials will be transmitted by the vestibular nerve into the brainstem in the cerebellum. Now, if someone tilts their head, and then that's towards the long stereocilia, as the stereocilia will be the shorter ones, the kinocilium will be the longest. So as the stereocilia bend towards this kinocilium, these hair cells depolarize and releases the neurotransmitter, which in this case is going to be glutamate. So they release more glutamate, and then look at what happened to the action potentials. It increases in number. And then when you return to the normal position, then there is no more bending of the stereocilia toward the kinocilium. So then you return to the normal levels of glutamate. And then your action potentials will be more spaced. And then what happens if you tilt the other way? So as the stereocilia bends away from the longest cilia, which is the kinocilium, these cells will hyperpolarize, they will release less glutamate, and then you have less action potentials. So that's what happens. So then how does the action potentials lead to homeostasis? Well, in several ways they contribute to homeostasis. For instance, part of the function of the vestibulocochlear nerve is to help us to maintain balance. If we maintain balance, we don't fall, we don't break our bones. That helps us to maintain homeostasis. In terms of food intake, well, I mean, with the special senses, we enjoy food, and then we can eat the food that we like, and that keep us healthy in a way, because I guess when someone will taste something, and if that taste doesn't appeal to the person won't eat it. So then we will end up with health problems. Another way that I can think about is, what if the food is spoiled? How would you know if you didn't have your sense of taste working well? So you can prevent that, and also by smelling. If you have some leftovers in the fridge, and then you had it for a long time, like a week, and then you were kind of lazy during that week, you didn't cook, and then you try to eat your leftovers from 1 week, and then you open your to-go box, and then you smells bad. I mean, would you eat it? No. So it helps us then to maintain health, the special senses. And also, it helps us to adapt to the environment and help to maintain internal stability. Now let's talk about some clinical points about the special senses, especially for the ear, like otitis media. Otitis media is inflammation of the middle ear, and typically it is caused by bacteria, but sometimes viruses also can infect the middle ear. That leads to pain, buildup of fluid, and hearing issues. So in terms of who is more prone or who's more likely to get a middle ear infection, well, typically it's children, and there is a reason for this, and under the age of 5. In children, the pharyngotympanic tube, the tube that connects the middle ear to the pharynx, is short and is horizontal. In adults, this pharyngotympanic tube is long and slender. It runs in an angle. That makes bacteria to travel less likely to the middle ear in adults than in children. And then around 90% of the children will experience an episode of otitis media during their lifetime. So that it is kind of high percentage, and typically otitis media is preceded by an infection of the throat. Now, then something important about this is that it is important for the child to have appropriate treatment and early detection, because that can prevent a complication in which the otitis media can go and infect this bacteria, or this microorganism can go and infect the mastoid process in the temporal bone. And then if that happens, don't think that that's a matter of antibiotics. Typically, it requires surgery. Now, when a kid has repetitive cases of otitis media, and sometimes it can be because they have very narrow pharyngotympanic tube, they can end up with an insertion of a tube that goes to the tympanic tube. So to help to prevent the accumulation of fluid. Then if you want to know more about this, you can go into that link. So this is a fundoscopic view with, this is a view of the tympanic membrane with an otoscope. So this is how this membrane looks like. This is part of the malleus look through the tympanic membrane. And then in here, we have acute otitis media. So it's reddish. That tells you that there is inflammation in there. And here we have a tube inserted into the tympanic membrane. This is called myringotomy to help to drain any fluids that has been accumulated there. In the next chapter, the next semester, you will learn in the endocrine system the different glands that we have in the body, how they communicate, and what is the function of all these hormones that we produce. You're going to learn the mechanisms and feedback loops between the different hormones. Now your test your knowledge questions. Which structure of the eye provides most of the eye's refractive power? Lens, cornea, retina, or iris? Remember, refraction is bending of the light. And in this case, towards the macula lutea, which is the area where we have more photoreceptors. So which of these four structures is the one that brings more light into the retina? A, B, C, or D?
Because it goes through the cornea, right?
Mm-hmm.
Past the iris. The back of the eye is the retina.
The retina.
So it would be the retina.
Well, the retina is the one that receives the light, so that doesn't.
And it bounces back up.
Not necessarily. What brings 2/3 of the light into the retina? The cornea. And then the other third will be by the lens. And then which cranial nerve carries the information of taste from the two anterior thirds of our tongue? 5, 7, 9, or 10? 7. The facial. And the posterior third will be? It's 2 out of the ones that we have there. Sensation of taste in the posterior third. 9 and 10, right? And what about pain? What if you bite your tongue and if you pain? What nerve carries that information? Or if you eat spicy food? You don't bite your tongue and say, oh, how good I taste. 5, trigeminal. True or false, the cochlea is involved in hearing and balance. True. It's one or the other is false. In which one is involved?
It involves in hearing.
Only hearing. So that's why it's false. So it will be involving hearing. True or false, rods detect color vision best in bright light.
Isn't it rods for color or cones for
CC, cones for color.
color? Cones for color.
Then
So that's
it is false. Rods is for black and white, grayish tones. And that's for nighttime vision. True or false, the vestibular system helps maintain balance and posture. True. True. And then describe the pathway of olfactory signals from the nose to the brain. So the olfactory nerve that starts in the nasal cavity in the roof basically extends axon that passes through what bone? Tiny bone that we have here.
Really gross.
That's the optic chiasma, right? Olfactory nerves, cranial nerve number 1 passes the axons through what plate of what bone? The one above the vomer. Ethmoid, the cruciform plate of the ethmoid, and then connects to what structure that looks like this, that you saw in the brain that you dissected, under under the frontal lobe, the olfactory bulb. Then connects to the olfactory bulb, and then you have axons of the olfactory bulb going into the Does it go directly to the olfactory cortex, or does it relay the information to the thalamus?
Well, it doesn't always involve the thalamus, does it?
No, it doesn't.
Because you can bypass it.
Yeah, it bypasses the thalamus. Basically, we don't ignore things that we smell. That's how it is. Okay, so from there it goes into the olfactory cortex. So that's how it works. And then also goes to the limbic system where we create memories of what we smell. That's all for